In this video, we're going to look at 5 different environmental factors that each affect how well an enzyme is able to catalyze its assigned reaction. The first of which that we're going to look at is temperature, because it's relatively straightforward to study as long as we understand the effects of temperature on protein folding. Remember that temperature is simply a representation of the average kinetic energy of a given chemical reaction system. The higher the temperature is, the higher the kinetic energy is, which means that the faster particles, including both the substrate and the enzyme itself, move within the reaction system. Now, the general pattern that we observe is that increasing temperature is also going to cause a corresponding increase in reaction rates, because the faster particles are moving, the more likely the substrate and the enzyme's active site are to collide with each other and undergo a successful reaction. Now, the converse is also true, that decreasing the temperature again causes a decrease in the kinetic energy of substrate and enzyme particles, which means that it is much less likely for the two to collide with each other and undergo a successful chemical reaction. Now, unlike uncatalyzed reactions, because enzymes are primarily composed of protein, we cannot just keep increasing the temperature forever and ever and ever, because very high temperatures will essentially cause the folding of the enzymes to shake itself apart, because the individual R groups on the protein are moving too quickly to have stable intermolecular forces. And this, of course, is what we call denaturation. Now, how high is very high depends on the organism. There are some organisms that can actually survive in nearly 100-degree water, but for humans, this normally begins to occur when your body temperature gets higher than 40 degrees. This would be an emergency situation where you would need to be rushed to hospital. Now, what we can see on this graph is that there is a point at which enzyme function peaks, and for most people, this exists around 37 degrees Celsius, which should surprise nobody, because this is generally regarded as the average body temperature, and this is what we call the optimal or optimally operating temperature for an enzyme. The next variable we're going to look at is pH, because the effects of pH are relatively predictable and easy to understand with respect to protein folding. Now, if we consider this protein to be a pretty generic one that basically floats around either in the cytoplasm or in your plasma, the liquid part of your blood, it would be surrounded by water, which we know has a pH of around 7, exactly 7 if the water is pure. That means that predictably we would find the polar R groups of all of these amino acids on the outside of our enzyme structure. Now, all enzymes require a very specific concentration of acid and a very specific concentration of base, because these are the two factors that affect their pH. So like temperature, all enzymes operate best at a specific concentration of acid and base, and this is what we call the optimal pH. And we can see for most enzymes that this number is right around 7. It's actually a little bit higher than 7, because there are a number of environments in the body that are just slightly basic. Now, if this enzyme does not exist in a solution that matches its optimal pH, which means either the concentration of hydronium or the concentration of hydroxide is different than its optimal pH, this is going to completely change the way that the enzyme folds, specifically the way that the enzyme's tertiary structures fold, because tertiary structure is determined by the placement of R groups. Now, if we introduce this to a lot of hydronium, we would expect the negatively charged acidic R groups to fold towards the outside of the protein in order to make ionic interactions with hydronium, and this would completely change the way that the enzyme forms. Now, for enzymes that are most used to existing in a pH of 7, if we introduce them to strongly acidic or basic environments, this will most likely cause the enzyme to denature as it loses its proper folding. An example of a very normal protein is hemoglobin, which exists in blood, which has a pH just over 7, and we see that reflected in the optimal pH of hemoglobin being between 7 and 8. However, there are extreme examples such as pepsin, which is the enzyme found in the stomach, which, as we know, has a very large concentration of hydrochloric acid, and therefore the optimal pH of pepsin is actually somewhere in between a pH of 0 and 2. The next variable that we can look at is the concentration of substrate molecules relative to the enzyme. Now, remember that the substrate is just the reactant that reacts with the enzyme in the catalyzed reaction. So logically, the more reactant we would have, we would expect the enzyme function to increase, or the rate at which enzymes could function to increase, and that's exactly what we see. So in a low substrate concentration, here we only have 4 substrate molecules relative to these 3 enzymes, and if these substrate molecules are not moving in the direction of the active sites, no matter what we do, they're never going to enter. But we can increase the probability of substrates being able to enter the active site if we just increase the amount of substrate that we have. The more substrate we have, that means that we have a better chance at the substrate entering the enzyme's active site and therefore forming the enzyme substrate complex, which will allow the enzyme to actually do the catalysis. And that's exactly what we see. Here we can see that all of the enzyme we have is actually filled up with substrate here, with tons of substrate outside to spare. Now, what this shows us is that we cannot keep increasing the substrate concentration indefinitely, because eventually all of the enzyme's active sites are going to become full of substrate. And if this is true, no matter how much substrate we have, no new substrate can enter the active site if all of their active sites are full. So at this point, we say that our enzyme has become saturated, meaning that each active site has become full of substrate, which is why this maximum point that we see on a rate of catalysis versus substrate concentration graph, this is what we call the saturation point at which we can no longer insert substrate into the active site. Now, if we wanted to overcome this saturation point, there is a very simple solution, and that is just to make more enzyme. So here, if we only have 2 particular enzymes, hitting the saturation point is very easy to do. Therefore, the more enzymes we have, the more active sites we have on those enzymes, and the harder it is in turn for all of the enzymes to become saturated with substrate, because once again, the more active sites we have, the harder it is for every single one of them to become filled with substrate, meaning that increasing the amount of enzyme that we have is going to push this saturation point higher and allow the enzymes to work at even higher peak rates. Now, if you think about this, you might say that the body will want to make as many enzymes as possible for every single chemical reaction so that we can just keep doing all of our reactions all at once. But this would actually be a very bad idea. For one simple reason, Are all chemical reactions always needed to be done every single time? Well, that answer is obviously no. For example, the chemical reaction to take glucose and turn it into glycogen, we don't need to do that if we don't have any glucose in our body, so producing a lot of those enzymes would be a waste of the body's resources. But the most important reason why we don't do this is that enzymes are not the only substance that is made out of protein. Your body needs protein in order to make transport proteins like hemoglobin or aquaporin to transport water in and out of cells. Your body needs protein to make muscle. Your body needs protein to make certain hormones like insulin. And if your body uses all of its protein reserves to just make enzymes, you would be depriving the body of other protein that it needs for other valuable purposes. So the body is perpetually in a great balancing act of allocating resources. The final category that we're going to introduce but save until the next video is something that we call allosteric manipulation. So for those that don't recognize this prefix, allo is a prefix that means other. And in this context, the other of what we have is another site. So here we can see that the substrate roughly corresponds to this active site here, but if we were to try to jam this substrate into what we call an allosteric regulation site, this substrate would not fit at all. So therefore, we can consider an allosteric site an other active site, also called a secondary active site, and the job of this active site is to bind a different type of chemical, which is what we call an allosteric regulator. And depending on what the regulator is, it may have a variety of different effects on the functionality of the enzyme, and these effects are all caused by the allosteric regulator changing the conformation of the enzyme. And this is done in exactly the same way that the induced fit model tells us the substrate would work. For example, maybe the allosteric regulator site has a negatively charged R group right here, which would correspond to a positively charged R group on the regulator molecule. And when it enters the allosteric site, the formation of an ionic bond between this positive charge and this negative charge would disrupt the intermolecular forces that normally stabilize the allosteric site like this. And as these intermolecular forces are disrupted, that means that the folding of this protein is going to be altered in some way, which will eventually have some effect on the active site, and therefore will change how well the active site is able to function. As briefly mentioned, we are going to focus entirely on allosteric regulation in the next video because there are a number of different possibilities that can result through an allosteric regulator having certain effects.